Well, first of all, I am Daniella Bittencourt. I am from Embrapa in Brazil. I'm also collaborator in the National Institute of Science, Technology, and Synthetic Biology in Brazil, coordinated by my colleague, Dr. Libio Heck. And this work I have developed at JCVI. I spent some time there with this great team, missing all my friends. And it's a little bit different from the previous presentations, which were more computational analysis. But now, as John said earlier, the JCVI-Syn3, the minimal cell, are not capable of contaminate or to infect mammalian cells. So it's a perfect chassis for us to identify genes functions. And in this work, we focus more on the mechanism behind mycoplasma host pathogen interactions. So for that, we have used not only the minimal cell JCVI-Syn3, but also JCVI-Syn1, this first synthetic cell, and the intermediated cells that were created during genome minimization. So first we checked which one of those cells were capable of grow and to infect mammalian cells. So for that, we analyzed several synthetic cells that were built during genome minimization. And we inserted co-culture this minimal cell with HeLa cells and HEK cells, and also with the media, only DMN media. We spent one or just brand new ones to see if the nutrients present in the media was capable also to support the minimal cell survival. And as we were expecting, we didn't see any growth from any one of our minimized genome, but only for the JCVI-Syn1, which has mostly of the Mycoplasma strain, wild strain genes. So, but then, which were trying to figure out which genes are responsible for these characteristics, we compare which were the genes that were deleted from these minimized genomes. And interesting, for this strain here, Syn1F, it only is different. It's only different from JCVI-Syn1 from the absence of a specific 8 genes. So here, just for you to have an idea how we check for the cell growth. Well, first, we put the minimal cell, the synthetic cell, together with the mammalian cells and cultivated them for a period of up to 15 days. And here we just have samples taken from up to 7 days. And then we could see once we let them grow, just that we use like only a sample from the supernatant of the medium in the culture bottle and add in the SP4 media, which is specific for the mycoplasma growth. And we observed, and then through the changing pH of the media, showing the cell growth, you can see which were the ones that were present in the mammalian culture according within time. So that's how we identified these strains that were capable of growing in mammalian cells. And then the difference between JCVI1 and JCVI1F, as I was saying, was these 8 genes. Interesting, the genes that we identified are two of them here are lipoproteins, which are well known to have a specific function between interactions between the mycoplasmas and eukaryotic cells, and also ABC transporters and other genes that are involved in metabolism of the cells. Interesting, this gene here, 183, it's a maltose ABC transporter. And recently it was shown that this gene is also responsible for the persistence of Mycoplasma gallisepticum in birds, infected birds. So this was showing us that we were in the right direction. So another thing that we also checked was the capability of DHH60 cells, neutrophil-like cells, to phagocytose the minimal cell and the synthetic cell Syn1. Here in the left, you can see a video. Oh gosh, I can see the video. Let me see something, just a moment. Okay, now we can. So here you can see the video of these bigger cells are the neutrophils, and they are moving afterwards the minimal, the synthetic cell, the Syn1. You can see that they are very active here in this nanolive video. When you check for this, the minimal cell, the phagocytic cells are not as active as we see in the previous, in the left video with the Syn1 cells, showing also that there is difference for the induction of phagocytosis. So we decided to check this characteristic too within the cells, and for that we move forward to add these genes back to the Syn3B cells. So the first we tried the lipoproteins. One of each we inserted back to Syn3B and analyzed if they were capable of surviving in cow culture with mammalian cells. And none of these genes were capable of staying alive with the mammalian cells. Different as you see here from Syn1, which we have all the controls, all the samples were grown very well. 179, 180, and 181 genes weren't able to recover this characteristic only by themselves. And here we have our controls in Syn3, which we don't see much growth of the cell after 7 days of cow culture. So then to check our species of these genes, we decided to insert them back in clusters. We did 2 different constructions. One that we add the 179, 180, and 181 genes. And the other construction, we add the 8 gene cluster all together. And we also inserted an emy gene, so we could be able to see the minimal cell attached with the mammalian cell. Then we move for the effectivity assay, where we co-culture in this slides chamber. We culture the HeLa cells, and then for 24 hours, then we add our synthetic cells, each one of them, and co-culture together with the HeLa cells 24 and 80 hours. And then we evaluated the DNA fluorescence, emy fluorescence, and also for oxidative stress fluorescence using cell rocks reagent. Okay, cool. So this, sorry, I think my computer is kind of slow. Okay. So this is just an overall picture of all the samples that we analyzed. So here we have the HeLa cells by themselves. Here we have for emy emission, for DNA using blue reagent, oxidative stress using the cell rocks reagent, and just this is the merged figure of all the treatment. So here just HeLa cells with no synthetic cell. These are the Syn1, the Syn3B with 179 up to 186 genes added back. Only the 3 lipoproteins added back, and this is Syn3 only with expressing emy. So just looking in an overall picture, you can see that we have much more emy emission in the Syn1, which was expected, and also with our added back with the 8 cluster of genes. So just to take a better close-up from the pictures, here we have our control, only HeLa cells, showing no fluorescence. Then here we have Syn1. If you see here in the right corner, you can see these small red dots. They are all the minimal cell attached to the HeLa cells. Similarly, we can see that with the add-back mutant, which we added the 8 genes together, showing also very high ROS activity here. See, you can also see the small red dots around the cells. We can see them in 24 hours after incubation and also 48 hours after incubation, showing that these genes are capable to make them the synthetic cell to the minimal cell to adhere to the mammalian cells. Differently, the Syn3B with only with the 3 lipoproteins together did not show the same characteristics of the other mutant with the
Danny,
age.
you need to wrap up.
Okay, so, and which is also interesting because showing that the ABC transporters are also important for these characteristics. So here we have the Syn3M cherry again, showing also after 48 hours, no presence of Syn3M cherry. And looking at the CCU analysis, the color change units, although the mutants were capable of attached to the mammalian cell, they are not capable to survive as long as the minimal cell. So it didn't recover the capacity of the synthetic cell to infect mammalian cells. We also checked for the flow cytometry analysis, looking at these cells cultured with DHL cells through time. And we also noticed a slight increase of phagocytosis activity in the add-back mutant with the A genes back. So also showing that these genes are important for the virulence of mycoplasmas. So which were the conclusions that we can take from these? Well, although we were not able to recover the capacity of the mycoplasma, the minimal cell, to infect mammalian cells, we were able to show that these genes are important for the adherence of the mycoplasmas to the mammalian cells. It's important also to point that JCV1, that the strain JCVI-Syn1 was exactly the same as JCVI-Syn1. Syn1F was exactly the same genome as Syn1, except for this A gene. So these genes might play very important roles for the infectivity of mycoplasmas in mammalian cells, even though they are not the only responsible for that. So we are keep going and check for other genes and try to identify exactly how this mechanism happened in mycoplasmas, which seems to be a very complex system for the infectivity of mammalian cells. Thank you very much, and sorry for the time, John.
So time for one question. Anyone?
Daniela, James Pelletier here. Beautiful talk. I'm curious if the reactive oxygen species is a general response of HeLa cells to being infected, or if that's something the mycoplasmas are bringing with them and inducing ROS somehow within the cells.
Yeah, well, actually, it's a mechanism. It induces the production for the ROS from the cells. So the HeLa cells start producing in response to mycoplasma infection.
Thank you very much.
You're welcome.
And James, the mycoplasmas, so the wild type mycoplasma does produce peroxide.
Yeah.
Not to be a virulence factor. But the minimal cells shouldn't.
Okay, thank you.
But actually, 1.0, we tried, well, we may have eliminated that capacity, but anyway.